Hello friends, in this video we will discuss germination of zygote in Oedogonium. We know that after fertilization, this is oogonia, antherozoid will come into oogonia through this spore, that is receptive spore, and it will fertilize this egg. Now this is zygote. So how this zygote further germinate and forms a new filament? In this video, we will study this process. The process by which it germinate and forms new filament. So this zygote, after fertilization, this zygote secretes thick wall around itself. First, it will secrete thick wall around itself. Then this oogonial wall will dissolve. This oogonial wall will dissolve and oogonial wall will dissolve and zygote will comes out in water. After coming out in water, it will settle down at bottom and it will rest there. After resting for some time, its protoplasm will divide by meiosis. Its protoplasm of zygote, this protoplasm will divide by, or you can say this cell, this zygotic cell will divide by meiosis and forms four small cells. So these are four small haploid cells. Four small it will, after division, after meiosis, it will form four haploid cell. Now each haploid cell will separate from each other, and at the anterior position of each cell, a row of flagella will arise. Now each cell will separate and a row at the interior end, a row of flagella will arise at each cell. Now these four are haploid spores, and these spores have flagella. So these spores will be called zoospores, and these zoospores are formed by meiosis. That is why they will be called meiospore. So what are meiospore of Oedogonium? Meiospore of Oedogonium is a haploid rounded cell like zoospore of asexual reproduction, and it have crown, or you can say row of flagella. It is similar to zoospore of asexual reproduction. The difference is, the sole difference between this spore and zoospore is these spores are produced by meiosis. That is why they are called meiospores. So these meiospores, after formation of zoospore, after maturation of these meiospores come out by dissolution of zygotic wall, and they will come out as vesicle, in the form of vesicle. So this is vesicle. They will come out in the form of vesicle. Soon vesicle will dissolve, and then after dissolution of vesicle, they will come out in water. And when they will come out in water, they will swim for some time. Then they will settle down at any substratum that they will find. So they will settle down at substratum towards anterior end. and then this anterior end, this colorless end will act as basal cell or hold fast. This upper region then divide further and like this divide further and this form a complete filament. So first, after dissolution of oogonial wall, zygote will come out into the water. It will rest for some time. Then it will divide by meiosis. 4 smaller cells will form. Then each cell will separate from each other. Then after separating from each other, each cell will form a crown of flagella at its interior end. Now these are called spores because they can germinate. These are called spores and they have flagella. That is why they are called zoospores and they are formed by meiosis. So the complete name of these spores is meiospores. Spores, they can germinate and form new filament. Zoo, they are flagellated. Meo, formed by meiosis. So this meiospore is complete name of these spores. These spores then come out as a vesicle. Then vesicle will dissolve and they will germinate at any substratum and they will form a new filament. So this is germination of zygote in Oedogonium.